All right, here's scene one of this origami. I have my human cell. I put the little ribosome inside. Here's my coronavirus with its little spike proteins and its RNA genome. And then I added something to my model that you don't have. I just used a little random scrap of paper and I created what is called the ACE2 receptor. This is a receptor inside of your human cells, particularly in your lungs, that's usually used for cell-to-cell communication, for your cells to communicate with each other. So this is receive a little message. Oops. But what COVID has done is evolved to have this spike protein that fits into this ACE2 receptor perfectly. And that is how the COVID virus binds to your cell and then inserts its viral genome inside. And so the virus itself and its lipid envelope and capsid doesn't even enter the cell. It just inserts its RNA genome. And it does that by communicating with this ACE2 receptor saying, hey, I'm friendly and I'm going to let this RNA genome inside. And then the RNA genome gets read by a ribosome, just like normal translation. And your ribosome translates the RNA genome and makes all of the proteins and things to make the virus. So then here's a new virus. Your cell here has this little virus making factory. All right, the ribosome just keeps on reading the RNA and making more and more copies of the virus, which is not good because eventually your cell will be filled with this virus from the ribosome and other enzymes and things making all the different parts of the virus. Your cell will be filled with it, and then, like we've talked about, the virus will bust out, make the cell explode, and then these viruses could go attach to more of your cells and cause a whole big nasty lung infection or all the symptoms of coronavirus. So that is without vaccination or without a prior infection. Your body is unaware, it just comes in, attaches, and craziness happens.